So let's discuss a provincial paper. The scheme that they have provided for us. This is their scheme, marking scheme. According to that, let's see the essays. So the first one. List out the characteristics of meristematic cells. So they are living cells. Number one, they are living cells. They are isodiametric, which means they're roughly spherical. They have less intracellular spaces. So if you see the cells, they have very less intracellular spaces. They're structurally and functionally undifferentiated, a very important point. They're structurally and functionally undifferentiated. So they are ready to be differentiated later, right? When the time comes, they differentiate. They have a central nucleus. They have a dense cytoplasm. No permanent vacuoles. Sorry. So they don't have permanent vacuoles. So they have a dense cytoplasm. They have the ability to multiply. They're meristematic cells, so they have the ability to multiply, increase in number. There are a large number of mitochondria. So you have a lot of mitochondria. So those are the points. Actually, in the resource book, they have mentioned, right? All cells in meristematics, all cells in meristems have common characteristics. They are, number one, they are living cells. Number two, they are isodiametric. Number three, they are structurally and functionally undifferentiated. Number four, they have a central nucleus. Number five, they have a dense cytoplasm. Number six, they have the ability to multiply. So the last one is actually irrelevant. Okay, you don't have to mention that. But as they have given, there's no problem writing. But I don't think we have to mention that because what's in the resource book, that's enough for this question. The characteristics of meristematic cells. Classify meristems according to the location. What do we have to do? We have to classify meristems according to the location. And then we have to describe the primary growth of a plant stem. Right. There are three types of meristems. There are three types of meristems. So what are the three types of meristems? Apical meristems, lateral meristems, and intercalary meristems. So apical meristems. So where are they located? These meristems, right? These meristems are located at root tips. Located at root tips and shoot tips. So where do we have them? We have them in root tips and we have them in shoot tips. Lateral meristems, where are they found? They are found in woody plants. No, they're found in woody plants. Now lateral meristems, right now, before that I have to mention, I'm going along with the resource book also. The apical meristems, what they have mentioned in the resource book is this. These meristems are located at root tips and shoot tips. We saw that in the marking scheme. They add new cells that enable the increase in length, right? They add new cells that enable the increase in length. Now that they have named that process as primary growth. Now we'll see how the marking scheme goes. Right, then lateral meristems, they are found in woody plants. Lateral meristems are of two types. No, what are they? Vascular cambium and cork cambium. Lateral meristems are of two types. They are the vascular cambium and the cork cambium. Very easy facts. Now what they have mentioned in the resource book about lateral meristems is as follows. Vascular cambium and cork cambium are lateral meristems. They are found in woody plants and also involve in the secondary growth in increasing the circumference of the roots and stems. The vascular cambium produces secondary xylem and secondary phloem. The cork cambium produces thick and tough periderm, replacing our epidermis. So right, then we follow the marking scheme. Now read the question. Now I'm pointing out, now this marking scheme is correct exactly. I'm pointing out the additional facts because another essay can be asked around this. Now see, they have asked us to classify meristems according to the location. That's all. Right? We have to classify meristems according to the location. That's the main criteria. Right? That's why they have described the location of apical meristems, the lateral meristem locations, not their functions, right? If they ask the functions, you have to write the functions. Right. Now, vascular cambium is one cell layer and consists of a continuous cylinder located between the primary xylem and the primary phloem, right? So the vascular cambium is one cell layer, right? It's just a cylinder of one cell layer. Consists of a continuous cylinder located between the primary xylem and the primary phloem. Cork cambium is made up of cells that arises in the outer layer of the cortex in stem. Cork cambium is made up of cells that arise in the outer layer of the cortex in the stem and in the outer layer of the pericycle in the root. Very important points these are. Cork cambium made up of cells that arise in the outer layer of the cortex, where in our stems, and in the outer layer of the pericycle in the roots. So the outer layer of the pericycle in roots, cork cambium is made up of cells that arise in the outer layer of the cortex in the stem and in the outer layer of the pericycle in the root. Now intercalary meristems, the next one. They are located at the bases of the stem and the leaves. Intercalary meristems are located at the bases of the stems and the leaves. Now, due to the activity of primary meristem in the root, primary growth of shoot takes place. Due to the activity of the primary meristem in the shoot, primary growth of the shoot takes place. So what? Due to the activity of what? Due to the activity of the primary meristem in the shoot, primary growth of the shoot takes place. Do you see how clearly in the marking scheme they have nicely mentioned the locations of the lateral meristems, intercalary meristems, and apical meristems? That's all that's required, right? How they are arranged, right? The location. When you're mentioning the location, you have to say how they are arranged also, no? You saw that clearly. Right? We saw the cork cambium is made up of cells that arises in the outer layer of the cortex in the stem and in the outer layer of the pericycle in the root. See how they are described? From that we can understand how they are arranged, how they are located, right? That's why you had mentioned those points. And in intercalary meristems, they have said that they are located at the bases of the stems and the leaves. Now we are coming to the next part of the question. So what's the next part of the question? They have said classify meristems according to the location and then describe the primary growth of a plant stem. Now we have to describe the primary growth of a plant stem. See how nicely they have come to the answer. Right, then this. Shoot apical meristem is a dome-shaped mass of dividing cells. Shoot apical meristem is a dome-shaped mass of dividing cells. So it's a mass which has this shape of a dome-shaped mass of dividing cells. They are dividing, right? They have the ability to divide. Where are they located? At the shoot tip. They are located at the shoot tip. Shoot apical meristem is a dome-shaped mass of dividing cells located at the shoot tip. Now, shoot apical meristem produces new cells only towards the stem. Very important point in MCQs. Shoot apical meristem produces new cells only towards the stem. So they produce new cells only towards the stem. But the root apical meristem, we'll see, it produces in both directions. Now, shoot apical meristem produces new cells only towards the stem due to mitosis. Now, by the elongation of these cells, what happens? Stem elongation takes place, simply increasing the height, right? We have to increase the height of the plant, the shoot. So, due to the elongation of these cells, stem elongation therefore occurs, takes place. This leads to the increasing of the height. Now it is the primary growth of the shoot. So this is what we call the primary growth of the shoot, right? Now this produces what? Primary tissues in the stem. So primary growth leads to producing primary tissues in the stem. Now in primary growth, right? In primary growth, three processes take place. And we're extending this discussion about primary growth. So in primary growth, there are three processes taking place. So what are the three processes? Number one, cell division. Number two, cell elongation. Number three, cell differentiation. So we have three processes. What are they? We have cell elongation, cell division, number one, then cell elongation, then number three, cell differentiation. These stages are found in three overlapping regions. Very important. These three stages are found in three overlapping regions. They are found within, right, three overlapping regions. They overlap, right? You can't see one as one separate unit. They're kind of mixed, right? So in certain regions, you can find both parts. That's the idea. In the zone of maturation, what happens? In the zone of maturation, the cells begin specializing in what? In structure and in function, right? So in the zone of maturation, so they are going to mature now. In the zone of maturation, what happens to the cells? The cells begin specializing in structure. Number one, structure. Number two, function. That's what we need, right? You need our cells. I'm not talking about our cells, I'm talking about the plant cells. We need our plant cells to specialize in structure and function. In the zone of maturation, that's why it matures there. Now, finger-like projections alongside of the apical meristems are called leaf primordia. Finger-like projections they are, right? They are finger-like projections alongside apical meristem. So they are called leaf primordia. So once again, the leaves, actually, what are the leaf primordia? They are finger-like projections along the sides of the apical meristem. Primordia cover the shoot apical meristem. They cover the shoot apical meristem. After cell elongation, what happens? Cell differentiation takes place. So after cell elongation, cell differentiation takes place. So once the cells elongate, right, increase in length, once the cells elongate, cell differentiation takes place following that. Okay, now some of y'all are very greedy on points, right? You say, well, I think this point should be included in the marking scheme. Even I have that means I feel, right, have they missed this point in the marking scheme? Should we include that? Will they cut marks or something like that, right? So let's see the resource book facts also along with this, right? So there's no confusion. If you really want, you can include those facts in the essay. So primary growth of the shoot, elongation of the shoot, I'm doing that fast because I've already discussed the marking scheme. Primary growth of this shoot. Elongation of the shoot is due to the activity of the primary meristem located in the shoot apex and is called the primary growth of the shoot. Elongation of the shoot is due to the activity of what? Of the primary meristem. Where is it located? Located in the shoot apex and is called the primary growth of the shoot. Now, a shoot apical meristem is a dome-shaped mass of dividing cells located at the shoot tip. So shoot apical meristem is a dome-shaped mass of dividing cells located at the shoot tip. Leaves develop from leaf primordia. So that point they did not include, right? Finger-like projections along the sides of the apical meristems. So leaf primordia are finger-like projections along the sides of the apical meristems. Now normally these primordia cover the shoot apical meristem. Shoot apical meristem produces new cells only towards the stem due to mitosis. Now, after cell elongation, cell differentiation takes place. Then the primary tissues of the stems are formed due to cell differentiation. That's a very important point, but they have not written that clearly, right? So you see, you all have to, in addition to reading the marking schemes, also follow the resource book. Resource book is the key, right? Resource book is the key because professors are making questions from the resource book. They are coining the answers using this resource book. You can't go beyond the resource book most often because they have given us the resource book now. From that only we have to find the answers. That's more than sufficient actually. So they have to say this clearly, right? Once again, what's that? After cell elongation, cell differentiation takes place. Now then the primary tissues of the stem are formed due to the cell differentiation. Now therefore the height of the stem is increased due to primary growth. So they have given the next question, describe the fine structure of chloroplast. The fine structure of chloroplast. So actually they have given the names in Sinhala, but you can go to your resource book and then in page 34 you can see the image. They have shown the intermembrane space that you can see, right? Intermembrane space, this region, intermembrane space. Then inner membrane, pretty easy, right? Inner membrane. Then the outer membrane, right? Then we have the granum. Can you see the granum? This is the granum, right? Then we have the free ribosomes. So free ribosomes. Can you see them? Free ribosomes. Then what else do we have? We have the thylakoid, right? Go to your resource book and see the thylakoid, right? There's one sac here, thylakoid, this part. Sorry, this part, I can't clearly show that. Just one thylakoid. Then we have the stroma. This is the stroma. We have DNA also. We have a cyclic DNA. Chakriya means cyclic DNA. That's the DNA there they have shown. Then we have intergranal lamellae, right? We have intergranal lamellae. They have shown that intergranal lamellae. Antargrana sustirka. Intergranal lamellae. Antargrana sustir, yeah. So that is the intergranal lamellae. So those are the basic things that you have to name. Go to resource book and look at that again. So we have to describe the fine structure of the chloroplast. Now it is a biconvex lens shaped organelle. It's a biconvex lens shaped organelle. It's a biconvex lens shaped organelle. Now with two membranes as the inner and the outer double membrane bound organelle. So it's a double membrane bound organelle because it has two membranes as the inner and the outer. It's a double membrane bound organelle. The outer and the inner membranes are smooth and are separated by a very narrow intermembrane space. These are very simple. I'm not going to explain a lot. Next point. Inside the chloroplast, there is a membrane system inside, right? Inside the chloroplast, what do we have? There's a membrane system with flattened, it should be flattened, okay? Flattened and interconnected sacs called thylakoids. So inside the chloroplast, what do you find? There is a membrane system with flattened and interconnected sacs. They are interconnected also, you saw that, right? In the image, go back to resource book and see this clearly. Called thylakoids. Now thylakoids contain complexes What do the contain? They contain complexes. What are those complexes called? They are called photosystems, and which are made up of what? Photosynthetic pigments, right? So thylakoids contain complexes called photosystems, which are made up of photosynthetic pigments. All right. Thylakoids are stacked. They pile on each other. They're stacked. Thylakoids are stacked to form a granum. We saw that in the image, right? Thylakoids are stacked to form a granum. The grana are interconnected by what? By intergranal lamellae. We saw that earlier, right? Thylakoids are stacked to form a granum. The grana are interconnected by intergranal lamellae. Now, stoma contains circular DNA, 78 ribosomes, many enzymes, starch granules, and lipid droplets. Thylakoids are stacked to form a granum. The grana are interconnected by intergranal lamellae. Stoma contains circular DNA, 78 ribosomes, many enzymes, starch granules, and lipid droplets. So the next essay, explain the process of converting what? Converting atmospheric CO2 molecule into a starch molecule in the process of photosynthesis in a C3 plant. So explain the process of converting atmospheric CO2 molecule into a starch molecule in the process of photosynthesis in a C3 plant. Now first, atmospheric CO2 enters into a leaf through stomata. Then CO2 is dissolved in the surface moisture of leaf mesophyll cells. That's nice, right? Then the CO2, they are dissolved in the surface moisture of the leaf mesophyll cells. And what happens? And then they are diffused into the leaf through the cell membrane. So then they diffuse into the leaf through the cell membrane, right? Then they can diffuse through the cell membrane. So first, you know what happens? Atmospheric CO2 enters into the leaf through the avenue of stomata. Then the CO2 is dissolved in the surface moisture, in whatever the moisture of the leaf's mesophyll cells, and then they are diffused into the leaf through the cell membrane. Now the palisade parenchyma and the spongy parenchyma are mesophyll cells. So palisade parenchyma and the spongy parenchyma are mesophyll cells. Then CO2 enters into the stroma of the chloroplast. CO2 enters into the stroma of chloroplast. Of what? Of mesophyll cells, right? We saw that. CO2 enters into the stroma of the chloroplast of the mesophyll cells. Now we saw that they pierced into the mesophyll cells, then within the mesophyll cells they enter the stomata. They enter the chloroplast. So CO2 enters into the stroma of chloroplast. Once they, after reaching the mesophyll cells, they get into the chloroplast. In which location of chloroplast? Into the stroma. 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 Energy from ATP and NADPH. So energy from ATP and NADPH produced by the light reaction are used to reduce CO2. So we need to reduce CO2. How? Energy from ATP and NADPH. Now, how are these molecules produced, ATP and NADPH? You know the light reaction. Yeah, from the light reaction. Now, what do they use these for? To reduce CO2. They have to reduce CO2. Now, the reactions are catalyzed by enzymes, right? We know most reactions are like that in plants, right? The reactions are catalyzed by enzymes. Catalyzed by enzymes. Now, the series of reactions take place in stroma. The series of reactions that take place in stroma in stroma is known as the Calvin cycle. The series of reactions that take place in the stroma is known as the Calvin cycle. Now it takes place in 3 steps. Now what are the 3 steps that it takes place in? 3 steps. Carboxylation, number 1. Reduction, number 2. Regeneration of RUBP. Regeneration of RUBP should be RUBP, that is ribulose bisphosphate. 5 carbon sugar, right? The carbon acceptor, you know that. So regeneration of RUBP, the carbon dioxide acceptor, you can say. Right, the first process is carboxylation. This is number 3. So the second process is reduction. The third process is regeneration of the carbon dioxide acceptor, or RUBP. For carboxylation, you can also say carbon fixation. Remember that. Right, so in carboxylation, the 5 carbon compound RUBP, in carboxylation, what happens is this. In carboxylation, 5 carbon compound RUBP accepts CO2. The enzyme involves in this reaction, the enzyme that involves in this reaction is RUBP carboxylase. The enzyme that involves in this reaction is our RUBP carboxylase oxygenase. I'm sorry, Rubisco, right? So the enzyme that involves in the reaction is RUBP carboxylase oxygenase, which is also called Rubisco. You know Rubisco. Rubisco. RUBP carboxylase oxygenase. Right, the next point. The first product is an unstable compound, which is a 6 carbon compound. The first product is an unstable one. It's an unstable, can't see that clearly, but the word is unstable, right? The first product is a 6 carbon product which is unstable. And therefore, as it's unstable, what happens? It breaks down immediately. Into 2 molecules of 3-phosphoglycerate. It breaks down immediately into 2 molecules of 3-phosphoglycerate. 3-phosphoglycerate is 3-PGA. Sorry, 3-PGA. So it, what breaks down? The unstable, the first product is the unstable 6 carbon product. It breaks down immediately into 2 molecules of 3-phosphoglycerate. So once again, the first product is a 6 carbon unstable compound. It breaks down immediately into 2 molecules of 3-phosphoglycerate, 3-PGA. Now 3-phosphoglycerate molecule is then reduced to glyceraldehyde 3-phosphate. Right, 3-phosphoglycerate molecule is reduced into glyceraldehyde 3-phosphate, step by step. So the 3-phosphoglycerate molecule that we saw now is reduced actually into glyceraldehyde 3-phosphate, step by step. This occurs in the reduction phase. Now you understand. Now this 31st point, it occurs in what? This point occurs in the reduction phase. Right? 3-phosphoglycerate molecule is reduced to glyceraldehyde 3-phosphate through step by step. Actually, in this resource book, they have mentioned it in a different way. It's the same thing. 1,3-bisphosphoglycerate will be reduced to glyceraldehyde 3-phosphate step by step. They have not mentioned that as 3-PGA, but they have mentioned a step further. No, 1,3-bisphosphoglycerate will be reduced to glyceraldehyde 3-phosphate. So these are the errors that occur. These are the problems with the resource book actually, right? Not very clear, the facts, especially in photosynthesis in this part, not very clear. So let's follow the marking scheme and write whatever is in the resource book for your health to get marks in your essays. So what's in this resource book is this. I'm pointing out the confusing facts. Reduction phase, we have 1,3-bisphosphoglycerate will be reduced to glyceraldehyde 3-phosphate. Step by step. So better write that. No? Write 1,3-bisphosphoglycerate will be reduced to glyceraldehyde 3-phosphate step by step. Right, so then the enzyme catalyzed reactions utilize what? So NADPH and ATP from light reactions. So enzyme catalyzed reactions utilize what? They utilize NADPH and ATP from the light reactions, right? So the enzyme catalyzed reactions can utilize this for their purposes. So G3P will act as a precursor for carbohydrate synthesis. So G3P will act as a precursor for carbohydrate synthesis to synthesize glucose. In the third step, what happens is this. In the third step, we have RUBP. In the third step, RUBP is generated by part of G3P through a series of complex reactions. Now, I'm not pointing out this mechanism, how it happens. You all have studied in the classes. So I'm just describing the essay to make it easier for you all to remember. It's a very long process to describe. You can't describe that in 10 minutes. In the third step, RUBP is generated by a part of G3P through a series of complex reactions. Now in the resource book, they have just said like this. RUBP is generated by undergoing a series of complex reactions. This process uses energy from ATP generated in the light reaction. That's what they have said. This process uses energy from ATP generated in the light reaction. But they have said additionally, in the third step, RUBP is generated by a part of G3P through a series of complex reactions. Now, this process uses energy from the ATP generated in the light reaction. We saw that. The remaining G3P is converted into hexose sugar and later converted into starch.